Hello everyone, and welcome to episode 51. The previous episode explored plant hydration, how plants move water and sugar through their bodies to sustain their photosynthesis and their growth. Today I'm going to be talking about something similar. I'm going to be talking about this plant tissue growth from another angle. Instead of hydration, I'm going to explore plant nutrition. Plants don't just need water and sugar and sunlight. They need particular element nutrients to create all the molecules that they need for their bodies. Proteins and DNA all require nitrogen and phosphorus, and numerous other enzymes and biomolecules require potassium, magnesium, sulfur, chlorine, and other elements like that. These are the nutrients that plants need to produce nucleic acids for DNA and amino acids for proteins. It's what they need to produce functional chlorophyll molecules and all kinds of enzymes. In this episode, I'll be exploring the mechanisms through which plants extract nutrients from the soil. Where last episode focused on the roots and the shoots and the vascular tissue, stuff including the entire plant's body, today's episode will focus more or less just on the roots, and how the roots selectively absorb the nutrients they need from the ground. This will involve talking about both the roots on a cellular level, as well as talking about things outside of the roots, like the type of soil that the plant is growing in. The type of soil is important because every quality of the soil influences nutrient extraction, from the organic content in the soil, like the dead and decaying biomatter, the detritus, to the literal size of the mineral particles. When the roots extend through soil and absorb nutrients, these nutrients get dissolved in the water and they flow up the xylem, and the upward flow of water deposits the nutrients throughout the plant's tissues, like a vertical river depositing silt along the shores and riverbed. The most absorptive part of the root is in a short region below the meristem. Recall from episode 49 that the meristem is a group of stem cells that perpetually divide into daughter cells. As the root meristem moves deeper into the earth, the root cells it produces go through phases of division, elongation, and maturation. These phases occur in the youngest cells, immediately following the meristem, and the most absorptive part of the root is in the zone of maturation. These cells are freshly mature and they're ready to go, and they're also the first cells that have a layer of epidermis with root hairs. These root hairs increase the surface area, and thus the absorptive capacity of the root tissue, tremendously. So the meristem penetrates into a region of soil, and because of the insane surface area allowed by the root hairs, the cells in this zone of maturation quickly absorb all of the nutrients in the region of soil. The perpetual root meristem growth allows the absorptive region of the root to continue digging downwards, perpetually seeking out new, nutrient-rich regions of soil. I want to dive into this subject of nutrients by first describing a few soil types that roots typically grow in, so that you understand the environment and the ecological context that the roots have evolved in. Soil is what composes the surface layers of the Earth's crust, above the mineral bedrock and below the air. Soil is composed of tiny particles and flakes of rock, as well as organic materials like bits of roots or chunks of wood, dust from dead skin, hair, decaying, crushed up, dried plant matter, bones, and shells, all kinds of stuff. The rock particles themselves are made through geological processes, by wind and water eroding stone over long periods of time to produce dust and small little pebbles. Over short lengths of time, water can get into cracks in rock and freeze, and when the ice expands, it cracks the rock apart further. Roots can do something similar. Roots will crawl into cracks, and as they grow wider with secondary growth, the rocks can be pushed apart and fractured. Soil can be anything from gravels and large rocks to pebbles and sand to clays and silt. The texture of a soil is really important. Soil composed of large, loosely fitting rocks with a lot of air pockets between them or air voids, like a typical gravel or a clean sand. These are well aerated soils, but they're poorly hydrated. Water just flows right out of them. They don't have much holding capacity. Because of this, larger plants will have no problem snaking their roots into the ground because they can exploit all these air pockets and stuff. But this coarse soil it doesn't hold water very well, and so it's hard for plants to sustain themselves without dehydrating. Now, on the other end of the spectrum, you have clays, or clay soils, and these are the opposite. Clays are composed of a lot of really tiny particles, or fines, as they're called, fine particles, and these absorb and hold water really well. Although sometimes they absorb and hold water too well, because sometimes this can make the clay seal up like a thick mud, and as a result, they become this impenetrable clay-like block. They're hard for the roots to physically penetrate, they hold water really well, but the downside is that they're very poorly aerated. They're almost anoxic. The soil content has very low oxygen. This discourages root growth, because it's hard for the roots to get in, but it's also dangerously anoxic, so the roots can suffocate from a lack of oxygen. Of course, soils usually are some mixture of gravel, sand, silt, and clay. There's a soil type called loam that's an ideal mixture of these elements. Loam is well aerated, it's easily hydrated, but it also works decently to hold onto the water without drowning the roots or physically blocking out the roots. The molecular qualities of the ions in the soil greatly influence its capacity to yield nutrients to living root tissue. Soil pH is really important. This measures the quantity of hydrogen ions in the soil, which react with plant root tissue in various ways. For example, soil with a high pH is basic, or alkaline, and it has a low concentration of hydrogen ions. Alkaline soils typically exist near limestone deposits, because the calcium carbonate in limestone reacts with water from like rain or something, and it also reacts with hydrogen ions together to create bicarbonate ions. These carbonate ions are formed when the CO3 of the calcium carbonate bonds with a hydrogen ion. And so basically the hydrogen ion gets absorbed into the carbonate material and pulled out of the soil. Acidic soils are the opposite. They have a low pH with a high concentration of hydrogen atoms. The acidity is typically generated through biochemical processes, where acidic biomolecules are formed in, for example, the decay processes of local plants. If you look at evergreen plants with needles like conifers or spruce, these are evergreen because they don't lose their needles in the fall. They hang on to their needles over the winter, but instead the needles are just gradually shed throughout the year as the individual needle ages and dries out and falls off. And when this happens, when these needles accumulate on the ground, they rot and they release nitric acid and phosphoric acid and other organic acids. This, in turn, makes the soil a lot more acidic. So a lot of boreal forests have very acidic soil. Most plants typically do well in neutral soils, with a pH around 6 to 8, although some species in various parts of the world are adapted to thrive in more acidic or more alkaline soils. It's just whatever they evolved to get used to. Nutrients in the soil come in the form of ions, or lone atoms of elements heavier than hydrogen, polarized with an electric charge. Ions with a negative charge are called anions. These are readily dissolved in water, and as a result they can be easily absorbed by the root tissue as it absorbs water. The dissolved anions in the water enter the plant xylem and they get swept up in the perpetual current moving from the roots to the shoots. And so, while this is relatively simple and straightforward, there's a downside to it. Because the anions are easily dissolved in water, they can also be easily washed out of the soil by rain. This leaching effect can strip the topsoil of nutrients, and that makes it much harder for plants to grow there. Positively charged ions, on the other hand, are called cations. These cations are also readily dissolved in water, but they're less bioavailable than anion nutrients, because these cations are also attracted to negatively charged compounds in the soil. Organic material, for example, tends to be negatively charged, and it'll attract and hold on to these cations. Soils made of clay are also very negatively charged, and these will tightly cling to cations. Clay soils also hold onto water really well, and through hydrogen bonding, the water holds anions, and through polar attraction, These anions hold on to cations, typically in the form of salts like Na+, Cl- ions, which make sodium chloride, or table salt. This hydrogen bonding and polar attraction makes the cations really hard to pull away from the soils, so the roots often struggle to absorb them. There are soils that are rich in organics and clays, and thus rich in nutrients, but they can often be terrible for growing plants, because even though there's all these nutrients in the soil, the plants just can't get a hold of any of them. They can't pull them out of the soil. They can't resist or overcome these electronegative relationships, these chemical bonds. In acidic soils with a high concentration of hydrogen ions, all of these hydrogen ions can interact with cations to pull them off of the organic matter in the clays in the soil. The hydrogen ions are positively charged, as are cations, so the hydrogen ions are able to sneak in and bump off the larger ions and take their place against the negatively charged surfaces of the clay or the organic matter. This is called cation exchange, because the protons are like unbound ions. They're free ions in the soil, and the cations, which are bound to the clays, are stuck there. And so when you do this cation exchange, and the hydrogen ion then gets stuck to the clay soil, it allows the cation, the nutrient, to be free-floating in the soil, and thus bioavailable for plant roots to absorb. Depending on the acidity of the soil, this cation exchange can be enough to overcome the negative attraction and make these cations readily bioavailable in large quantities for the plants that grow there. All of these ion nutrients are scattered throughout the soil in a relatively low concentration. But inside the plant's tissues, the nutrients are tightly concentrated into enzymes and DNA and other biomolecules and stuff like that. What this means is that to get nutrients out of the soil, the nutrients have to be pulled against their concentration gradient. Consider that this is the opposite of water, which follows a gradient of decreasing water potential as it moves from the soil into the roots, up the stem, and out the leaves. So with regards to nutrients, how do plants do it? How do they go against the ion concentration gradient to absorb nutrients out of the soil? Long story short, the answer is proton pumps. Understand that the epidermal cells have plasma membranes partially exposed to the air. The root hairs are just projections of exposed cell membrane. They aren't protected behind a cell wall. Plant root cells and the epidermal cells with root hairs have enzymatic structures in their membranes called H+ ATPases, which are enzymes that consume a molecule of ATP or break it down, hence the ATPase, to push one proton or one hydrogen ion out of the cell and into the nearby soil. This energy-expensive process creates a pool of protons directly outside the root cell membrane. What effects do you think are caused by a high proton concentration? If you said low pH or high acidity conditions, you're correct. Additionally, the positive charge on all the protons creates a positive charge on the outside of the cell membrane. This creates a voltage difference between the inside and the outside of the cell, which creates an electrochemical membrane potential. So these proton pumps push a lot of protons out of the cell, and in addition to creating a proton concentration gradient, this makes the conditions outside the membrane acidic and positively charged, while conditions inside the membrane are negatively charged and less acidic. The positive charge accumulating outside the root tissue will repulse positively charged ions in the soil, like cations like calcium and potassium. And because the inside of the cell is negatively charged, this electrochemical gradient attracts the cations enough to pull them out of the soil and into the cell. The cations are too big to move through the sheet of phospholipids in the membrane itself, so instead they move through protein tubes, called channels. These protein channels penetrate the cell membrane and create a travel corridor for the cation. Because of the electrochemical gradient, it's all just a matter of the cation finding itself close enough to the appropriate channel to get absorbed into the cell. And next, we have anions, the negatively charged ions, which are actually repulsed by the negatively charged environment inside the cell. And so, to move the anions against their electrochemical gradient and get them into the cell where it's relatively negatively charged, the root tissue has to tap into the proton electrochemical gradient that it established earlier with the H+ ATPases. To do this, root cells use enzymes called cotransporters that can move two particles simultaneously. The direction each particle moves depends on what kind of cotransporter the enzyme is and what its purpose is. For example, a cotransporter enzyme that moves both particles in the same direction is a symporter, while an antiporter moves the particles in opposite directions. So for example, one particle comes in, the other goes out. Organisms use different cotransporters depending on their chemical needs. In plants, a symporter is used to transport a proton and an anion from the soil inside the cell. To do this, the symporter has to move the proton along with its concentration gradient, which is easy because the proton just wants to move right along with it. But it has to move the anion against the electrochemical gradient, and that isn't easy. However, because of the proton pumps and their perpetual regurgitation of protons back out into the nearby soil, the proton electrochemical gradient is really big. It's large enough so that the energy harvested from letting a proton in along its gradient is more than enough to push an anion against its electrochemical gradient. So it's through the proton pumps that root cells are able to create the external conditions necessary to attract and simultaneously absorb the positively and negatively charged ion nutrients that exist out there in the soil. But there are other cations and anions in the soil, ions less conducive to the good health and nutrition of the plant. This might include metal ions like cadmium, lead, and zinc, which can all be harmful to plants by disrupting their enzymes and gracelessly interrupting the biochemical orchestra that goes on inside their cells. Even nutrients that the plant legitimately needs, like magnesium or calcium, can be dangerous if the plant is exposed to too much of it. First and foremost, the root cells have a passive defense system that protects them from particular ions. This passive defense system is basically the lack of specific enzymes. If the cell lacks the genes to make a protein channel or a cotransporter that would allow cadmium to come into the cells, for example, then the cell simply won't express any transport enzymes that can move cadmium, and cadmium will thus not be able to enter the cell in the first place. That's pretty simple. But other ions can sneak around this by flowing in the water going in the apoplastic pathway between the cell walls. But these ions are stopped from entering the plant's vascular tissue by the Casparian strip. This structure, this Casparian strip, is a waterproof sheet of endodermis that creates a seal between the vascular tissue and the ground tissue. In cases where the plant cells do express a transport protein that allows certain ions in, these ions can still poison the plant if they become too concentrated. This can be a serious problem for plants that exist next to a mine, as they're inevitably exposed to soils rich in the mineral runoff of the mined-out earth. Zinc mines, for example, saturate the nearby soil with zinc particles, and the plants have to deal with this. Usually, the plant will take in the tiny amount of zinc that they need, but when they're exposed to it in high concentrations, like they are next to a zinc mine, it can build up in their tissues to dangerous levels. To prevent themselves from getting metal toxicity, the plant expresses genes that code for little proteins called metallothioneins. These itty-bitty metallothioneins will coat the metal ions and basically nerf them and make them unreactive and non-toxic. Remember from last episode how I talked about how roots can store sugars in their vacuoles? The vacuole is basically a huge, empty storage pocket inside of every plant cell, surrounded by a selective membrane called a tonoplast. Various plants have all kinds of proteins in their tonoplasts that will help regulate the flow of ions or particles or molecules or whatever is being put into the vacuole as storage. In this case, the tonoplasts have proteins that will suck up metal ions from the cytoplasm and store them safely inside the vacuole. This is an interesting defense, as the plant cells aren't actually removing the toxic amounts of metal from the plant's body. They're just isolating the metal ions in a safe place within their cells, kind of like an armored dumpster. When you look at the tissues of plants that are able to survive in metal-rich soils, you might see discolorations, as so much metal has been accumulated in all of the vacuoles of the plant cells that it's actually creating a visible shift in its outward hue, almost like pigments. Alright, so I've talked a lot about nutrients and ions and how the plant absorbs them, but I haven't really talked about what these nutrients actually are and what the plant uses them for. In the first 2 episodes of this series, I talked about how plants breathe in carbon dioxide and breathe out oxygen. And in the last episode, I talked about how plants absorb water and move that water through their body up to their leaves. But plants also need calcium, which exists as the ion Ca2+. It's a cation with a 2+ positive charge. The plant uses calcium for all manner of purposes. It's a part of the cell wall. It's used in enzymes. It stabilizes the plasma membrane. And it does a whole lot more. If the plant lacks calcium, it'll suffer catastrophically for it. Its roots and its leaves will be malformed, and they'll grow into weird shapes, and the whole plant will be riddled with necrotic patches of dead tissue. Another common nutrient is potassium, which has the ionic symbol K+, signifying that it has another positive charge. Potassium is used like calcium in all manner of regulatory functions and enzymatic processes. Potassium is really important for regulating water concentration and water transport through the plant cells. Without enough potassium, the plant's ability to regulate its water concentration is impaired, and you'll see that its growth will be diminutive and its tissue weak, because the tissue lacks the turgor pressure needed to keep it strong and upright. Water and carbon dioxide are critically important nutrients for plants, but calcium and potassium are also necessary nutrients too, as are sulfur, phosphorus, and magnesium. All of these are macronutrients, which are nutrients that the plant needs in relatively large quantities. Now, if there's macronutrients, that kind of implies that there's micronutrients, or nutrients that the plant needs in relatively small quantities. Some of these micronutrients include zinc, boron, copper, iron, and chlorine. Zinc is used for enzyme regulation and synthesis of a growth hormone called auxin. Boron is used to stabilize both cell membranes and cell walls. Copper is used in enzymatic reactions, and it's used in lignin, which is the main structural molecule in heavier plant tissues like wood. Iron is necessary for many functions, including the synthesis of chlorophyll, which the plant obviously needs to conduct photosynthesis. Chlorine is required for its interactions with water, including a critical step in photosynthesis, which requires breaking down water molecules. Deficiencies in any of these micronutrients will lead to subtle but increasingly obvious problems in the plant, like chlorosis and little spots of necrosis. One of the most important macronutrients that plants need is nitrogen. Nitrogen is used in almost everything in a plant, from its proteins and DNA to ATP to chlorophyll. Without enough nitrogen, plants will be weak, small, and their tissues yellow. They will be malnourished in every sense of the word, as they don't have a key chemical ingredient for virtually every protein and cellular function that they need to survive. This high demand for nitrogen has been a hugely motivating evolutionary force. In both plants and fungus, as well as bacteria, this need for nitrogen has promoted diverse mechanisms of nitrogen acquisition, like nitrogen fixation, cellular symbiosis, or even a carnivorous diet. Nitrogen is really common. In the soil, it exists in nitrate ions, or in nitrogenous compounds like ammonium. But plants need far more nitrogen than they can ever hope to get out of the soil. So it's good for them that nitrogen is also in the air, where it makes up almost 80% of the planet's atmosphere in the form of molecular nitrogen, or N2. But there's a catch. Just because you have all of this nitrogen there, doesn't mean you can access it. The thing about molecular nitrogen, about N2, is that these two nitrogen atoms are bound by a triple bond. That triple bond is really strong. It's really hard to break. Because of the inherent difficulty in breaking a triple bond, only a very small, select handful of species have ever evolved the chemical means to do it. And even within these species, the process of breaking down molecular nitrogen, which is called nitrogen fixation, is an extremely energy-intensive process and it requires a hugely complex suite of proteins all working together. Every species that can fix nitrogen is microscopic. They're bacteria and archaea. But this is good news, because it means that they can more easily live inside the cells of plants. This is exactly the case with bacteria in the genus Rhizobium, many of which reside inside the cells of plants in the legume family. Because of the nitrogen-fixing bacteria that extract molecular nitrogen from the air and convert it into bioavailable nitrogen sources, these legume plants are nitrogen-rich. Legumes like peas, soybean, and clover have little nub-like growths or bulges called nodules, and these nodules are filled with nitrogen-fixing bacteria. Understand, though, that it's not just an empty sac that's filled with bacteria. It's actually a region of plant cells that are infected with bacteria. Just like the tuber is a portion of root tissue with sugars packed into the vacuoles of the cells, the nodules in these legumes are plant tissue with cells packed with nitrogen-fixing bacteria. The cells create an internal environment devoid of oxygen, which would otherwise destabilize the proteins required for nitrogen fixation. Inside of these optimized plant cells, the rhizobia can safely fix nitrogen and feed what it produces back to the plant. It's really interesting how this symbiotic relationship happens. The legume plant releases chemicals out of its roots called flavonoids, which come into contact with the rhizobium bacteria and stimulate them to create nod factors. These nod factors are chemicals that come back into contact with the legume's root cells. The nod factor binds with certain proteins and activates a very specific signaling mechanism within the legume cells. Now this is where stuff gets really crazy, because this primes the root cells to create an infection thread. When a rhizobia bacteria touches a primed root hair, it creates an inward projection of the plasma membrane called an invagination. Where the root hair is an outward projection of the membrane, this inward projection is like a similarly shaped tunnel, except it goes back into the root hair itself. This tunnel extends down the root hair into the cortex, into the root tissue of the plant itself. The bacteria thrive in the opening to this tunnel, and when it descends into the plant itself, the bacteria flow down this tunnel into the plant cells. This tunnel is called an infection thread, because it's quite literally the thread by which the legume root cells are infected with rhizobium bacteria. Once infected, these cortex cells divide and create the outward growth called the nodule. All of the cells in this nodule are infected with rhizobia, although the word infected implies a negative relationship. Instead, the infection creates a symbiotic relationship shaped by evolution, where the bacteria produce and share nitrogenous compounds, and in return, the plant will produce sugar and share some of its sugar with the bacteria. Although, don't be mistaken and think that this symbiotic relationship is perfectly equal, because it's not. You could almost say that the plant is getting taken advantage of. I mean, sort of. The plant needs nitrogen. It needs nitrogen really badly, and the rhizobia can provide that nitrogen, but at a really high cost. The rhizobium bacteria require a huge amount of sugar to fulfill basically all of its needs. To grow and divide, it needs chemical energy, and sugar provides that chemical energy. It also needs carbon compounds to physically grow itself, and sugar provides that carbon. Then you have the process of nitrogen fixation, which is in and of itself very energy expensive. The plant has to provide all of the sugar that the rhizobium bacteria need to fix their nitrogen. And in return for this huge sugar investment, the bacteria will produce a little bit of nitrogen, which the plant rapidly gobbles up and incorporates into its proteins, its cells, and its DNA. It shouldn't be too surprising then that some plants look at this symbiotic relationship and say something like, "Forget that, I'm not being taken advantage of by some stupid bacteria." And that's all well and good, but these plants still need nitrogen. So if they aren't evolving a symbiotic relationship with nitrogen-fixing bacteria, then how do they get it? How do they get their nitrogen? Well, there are other sources of bioavailable nitrogen in their environment. Some plants are parasitic, and they'll grow on other plants and penetrate their tissues to tap into their xylem. And here they can steal water and nutrients directly out of their host plant. Some plants are less invasive. These are called epiphytes, and while they grow on other plants, they don't parasitize their hosts. Instead, epiphytes express morphologies that allow them to capture and retain airborne particles. Epiphytes have leaves that overlap in such a way so as to create little recessions or half-bowl shapes, and these can hold rainwater like a cup. They can also accumulate dust and pollen and other airborne particles, and all of these can be broken down and absorbed as food. There are also plant species that will develop symbiotic relationships with fungus. Lastly, and perhaps most interestingly, there are some plants that turn to meat for their nitrogen, like insects and even small vertebrates. But plants can't eat meat. They aren't carnivores, they don't have mouths. They just have roots and leaves that absorb stuff on a molecular level right out of the air, right? Well, not exactly. Some plants really are carnivores. They're carnivorous plants, and they do have a mouth, or a mouth-like structure, or something that works kind of like a mouth. Several lineages of these carnivorous plants have evolved mechanisms to capture insects and dissolve their bodies in a way that their nutrients can be absorbed. The capture mechanism is typically created out of leaf tissue that evolved a specialized purpose. In pitcher plants, leaf tissue evolved to form an enclosed basin, or a cup, which lures insects to their death. They land on the rim of the cup, or the pitcher, and because it's covered in this kind of slimy, slippery fluid, the insect will slip. They can't get a good purchase, they can't get a good foothold, and they fall inside the pitcher. At the bottom of the pitcher, they get soaked in a fluid solution, and this traps them, it ruins their wings, and it slowly dissolves them. This dissolved insect is then absorbed by glands at the bottom of the pitcher. In plants like the Venus flytraps, the leaf tissue has evolved into two sensitive plates, like jaws, and on the inside of each plate are a few little hairs. When an insect lands on the leaf plate and bumps into these sensitive little hairs, they send a signal that causes the leaf plates to slam shut and trap the insect. As the insect struggles and fights back inside the plant's grasp, they continue to hit and bump into these root hairs, and because these root hairs are continually stimulated, they encourage the secretion of digestive enzymes. So the captured insect struggles, it fights for its life, but this struggle literally causes the plant to release the acids and digestive chemicals that break down the insect and dissolve its body into goo. And then what's left of that nitrogen-rich goo gets absorbed by the plant. What's kind of interesting, and it makes ecological sense, is that these carnivorous plants are usually found in areas where nitrogen is rare in the soil. And I mean, this makes sense, as living in a habitat with poor nitrogen would encourage the plant, evolutionarily, to find new means of acquiring nitrogen. But studies have shown that if you grow these carnivorous plants in nitrogen-rich soils, they actually produce fewer of these insect-eating structures like flytraps and pitchers. In this way, the production of the carnivorous plant mouths seems to be regulated by genetic expression, and that genetic expression is itself regulated by the diet of the plant and the availability of nitrogen in the soil. If there's a lot of nitrogen, then the plant doesn't need to invest energy and nutrients in growing these pitchers or these fly traps. But if there isn't a lot of nitrogen, then the plant needs to start looking at alternative sources. It needs to start supplementing its diet with nitrogen. And to do that, it'll grow more pitchers and more Venus flytraps, or whatever mechanism it has. Well, that's pretty much all that I have for you today on plant nutrition. I hope you enjoyed this episode, and I hope you learned something cool about a plant's diet, something that you found interesting or fascinating. Maybe something about how plants extract nutrients from the soil, despite all the concentration gradients and electrochemical gradients that get in the way. It's fascinating how organisms and evolution can come up with all of these really complicated chemical structures that help it survive in various environments, like high acidity soils or low water soils or what have you. If you're enjoying this series on plant physiology, then stick around for the next episode on plant senses. I'll be discussing how plants maintain their internal homeostasis, and how they get a primitive awareness of their external conditions, like the direction of gravity, the location of the sun, and even the presence of herbivores eating their leaves. So if that sounds cool, then stick around and check it out. And as always, thanks for listening.
Would you like to support the BioLogic podcast? It's super easy. When you open a new episode, press the like button or share it with your friends. If you aren't subscribed, you should hit the subscribe button so you can enjoy a new episode every week. You can also peruse our official store, which has a ton of cool stuff like hand-designed t-shirts, hoodies, and stickers. All the links you need are in the description section below.